The diversity of life on Earth is astounding. Organisms both large and small have adapted to the many different habitats on the planet, and along the way, they have developed unique traits and methods of survival. But at a microscopic level, all living organisms are very similar. They are all made of cells. A single cell contains the information that determines all an organism's characteristics. A giant sequoia in California begins as a single cell and eventually grows to be one of the largest trees in the world. As the fundamental unit of life, the structure and function of cells dictate many of the basic behaviors of living organisms. Animals eat because their cells need energy, and they breathe because their cells need oxygen. Cells are able to function and thrive on their own. Unicellular organisms are so successful that they can be found in every habitat on Earth, no matter how extreme. Without cells, life as we know it would not exist. Life on Earth is diverse, but it all can be traced to a single building block, the cell.
All the requirements of life can be traced to what cells need to survive. By understanding the structure and function of cells and the differences between types of cells, we have a better understanding of the world around us. Living cells are formally categorized into two groups, prokaryotic and eukaryotic. But no matter how different, cells share some common characteristics. Every cell contains cytoplasm, a watery gel that makes up its bulk. The cytoplasm is composed of cytosol, the fluid component of cells, and any organelles. The cytoplasm circulates around the interior of the cell. A membrane surrounds the cytoplasm, composed mainly of two layers of phospholipids. The membrane is full of pores and channels that allow the cell to take in and expel various molecules. All cells contain genetic material in the form of DNA and ribosomes that play a vital role in the manufacture of proteins. Ribosomes are very small bead-like structures that can float in the cytoplasm or attach themselves to interior membranes. The location of the DNA is the primary difference between the two groups of cells. In a prokaryotic cell, the DNA is bunched together in a nucleoid region, but floats free in the cytoplasm. In a eukaryotic cell, the DNA is physically separated from the cytoplasm by a membrane. This structure is called the nucleus, and its presence is what determines whether a cell is prokaryotic or eukaryotic. Bacteria are the best known form of prokaryotic cells. Because they can live in a wide range of environments, they are one of the most widespread forms of life on the planet. Another form of prokaryotic cell is known as archaea. These prokaryotes differ from bacteria in the chemical composition of some cellular structures, and they are found mostly in extreme environments, including these hot springs in Yellowstone National Park. Prokaryotes are small enough that they can rely on diffusion alone to transport internal materials. Molecules in the cytoplasm float freely, and they will easily encounter other molecules to undergo reactions and keep the cell alive. Eukaryotes are much larger than prokaryotes, so they need more organization to stay alive. Eukaryotes have organelles, internal membrane-bound compartments. Each organelle serves a specific function in the cell, from molecule storage to energy production. The largest structure is the nucleus, which contains the cell's DNA. To fit inside the nucleus, the DNA is wrapped around proteins. One of the cell's primary functions is to manufacture proteins from the code contained in the DNA. First, the code is transcribed within the nucleus from the DNA into RNA. Then the RNA travels into the cytoplasm, where it encounters a ribosome. The ribosome translates the code in the RNA into a protein using amino acids floating in the cytoplasm. Some of these proteins get carried to their destination in the cell, and others enter the endoplasmic reticulum. Located right outside the nucleus, the endoplasmic reticulum is an organelle composed of a series of membranes. Proteins that are leaving the cell or going to the outer membrane are chemically modified here and sent to the Golgi apparatus. A series of flattened organelles, the Golgi apparatus further modifies the proteins and sends them to their final destinations. Protein production is the main function of many organelles, but not all of them. Eukaryotic cells contain microtubules and microfilaments that support the cell and provide pathways that allow molecules to move from one part of the cell to another. The division of labor in eukaryotic cells is the key to their survival, and this has allowed them to work together in larger multicellular organisms. Did you know? Scientists theorize that modern eukaryotes evolved from a symbiotic relationship between different prokaryotes. This theory states that some organelles were originally bacteria that entered other ancient cells, but did not cause infection and were not digested. Over time, this relationship became so ingrained that now larger cells can't survive without these organelles.
The structure of eukaryotic cells gives them incredible versatility. Eukaryotes can be unicellular or multicellular, plant or animal. This flexibility has led to an immense variety in the lifestyles and appearances of eukaryotic organisms. Organisms in the eukaryotic group are classified into more specific groups, namely plants, animals, fungi, and protists. The main difference between plants and animals lie in the source of their food. All cells use adenosine triphosphate, or ATP, as the primary source of chemical energy. And most eukaryotic cells create ATP in cellular respiration. Cellular respiration takes place mainly in the mitochondria, present in nearly all eukaryotic cells. In these organelles, a sugar called glucose combines with oxygen to create carbon dioxide, water, and energy. The energy is stored in molecules of ATP, which cells use to power many of the mechanisms that keep them alive. The difference between plant and animal cells is the source of their glucose. Animal cells are heterotrophic, meaning they need to absorb glucose from an outside source. Plant cells are autotrophic, meaning they make their own food by photosynthesis. This process is almost the reverse of cellular respiration. The cell uses energy from sunlight to convert carbon dioxide and water into glucose and oxygen. Almost all the atmospheric oxygen on the planet results from the process of photosynthesis. These different sources of food have led to some structural differences between plants and animals. Plant cells have a cell wall outside the cell membrane. The wall provides support, which is what keeps plants stiff and upright and allows them to grow without bending or breaking. This helps plants stay oriented towards the sun. Plant cells also contain a large central vacuole filled with water and other molecules. When the vacuole is full, the water pressure helps keep the plant stiff and upright. Plants wilt when they lack water because their vacuoles do not provide this added support. Plants also have an additional type of organelle known as a chloroplast. The location of photosynthesis, chloroplasts are the structures that provide food for the cell. They contain a green pigment called chlorophyll, which assists in photosynthesis and gives plants their color. Animal cells lack both a cell wall and chloroplasts. Without chloroplasts, heterotrophs must move around to find food sources. This difference in activity is evident at the multicellular level. Animals are mobile and plants are stationary. Because plant cells produce their own food, they are the basis of many food chains. Higher organisms in the chain get energy by ingesting the plant cells and their sugars. Even carnivores benefit from the plants by eating other animals that fed on plants. This is true for multicellular and unicellular eukaryotes. While most single-cell heterotrophs ingest molecules, the amoeba and some others actively hunt smaller cells. Did you know? Photosynthesis is the most common form of food production, but not the only one. Some archaea living in extremely harsh environments are chemosynthetic, meaning they use chemical compounds instead of sunlight to produce energy. In some environments, such as hot water vents miles beneath the surface of the ocean, whole ecosystems have developed that rely on these unique life forms for survival.
Though they are small, cells lead complicated lives. In addition to maintaining enough energy to power cellular processes, they must replicate themselves accurately. And cells in multicellular organisms must develop to carry out highly specific jobs. A main biological impulse among organisms is to reproduce. Living things spend time and energy to produce offspring and continue their species. The same is true for individual cells. Most cells reproduce asexually, meaning that they do not trade or combine DNA. The offspring of asexual reproduction is a clone or genetic replica of the parent cell. Most prokaryotic cells reproduce by a process called binary fission. The cell replicates its DNA so that it has two copies, and then it splits in half. Each new daughter cell has one copy of the parent DNA, and both are clones of the original cell. Eukaryotic reproduction is more complicated because the cells themselves are more complex. Eukaryotes must replicate their organelles as well as their DNA. Before a eukaryotic cell divides, it replicates its DNA so it has two complete copies. During mitosis, the parent cell arranges the two copies of DNA into matching pairs of chromosomes that are evenly split to form two new nuclei. When mitosis ends, cytokinesis begins. Cytokinesis is the process in which the cell's organelles and cytoplasm are split, and the parent cell physically separates into two new daughter cells. Most multicellular organisms rely on another type of reproduction, known as sexual reproduction, which involves the combination of two different sets of DNA to form a new, unique organism. Here, two unique types of cells called gametes combine. Gametes contain only half the number of chromosomes usually found in an organism. Human cells normally have 23 pairs of chromosomes, but human gamete cells contain 23 single chromosomes, also called chromatids. Gamete cells are the result of meiosis, during which a parent cell divides twice. The first division is very similar to mitosis, where the DNA is replicated and each resulting cell gets a full set of chromosomes. In the second division, the DNA is not replicated, and each chromosome is split in half, with the resulting cells having only half a set of DNA. The combination of two gametes into a new cell is called fertilization, and the cell is called an embryo. For example, humans start as embryos, and both plant seeds and bird eggs contain embryos. As an embryo develops, the cells need to differentiate. They take on characteristics specialized for specific functions. Muscle cells are long and elastic to provide strength and flexibility. Nerve cells have long extensions that let them connect with other nerve cells and send and receive messages rapidly. Red blood cells lack a nucleus, allowing them to squeeze through small capillaries. This ability of multicellular organisms to develop specialized cells that can communicate and function in a coordinated way has led to the development of the great variety of species of plants, animals, and fungi on our planet. Cells are the fundamental units of life, remarkable building blocks that create and sustain life in all of its forms.
What if you were so severely burned that you couldn't recognize your own face in the mirror? But a patch of skin made of discarded human cells could painlessly restore it to normal. Suppose you were a promising young athlete and the cartilage in your knee was damaged beyond repair. But with the help of a serum-based growth solution, it was possible to grow new cartilage. Or what if you needed a new liver, but instead of a transplant, doctors simply repaired your old one? All of this is becoming possible thanks to a new field of medicine called tissue engineering. The ultimate goal is to repair organs by growing healthy cells in a lab and injecting them into the body. Researchers are already growing blood vessels, cartilage, and bone, and even have off-the-shelf skin. The cells in this one tiny piece of skin can produce enough healthy skin to cover 5 football fields. And for burn patients like Catherine O'Leary, it's nothing short of a modern-day miracle. Catherine and her husband Cyril were sailing. The plan this particular weekend was to sail across the harbor from their home in Fairhaven, Massachusetts, drop anchor, and spend the night across the bay near Cape Cod. The next morning, just as she did every day on the boat, Catherine got up early to make the coffee.
I got up, I went to the galley, I got the coffee pot out, the coffee.
But when she turned on the stove and lit the match, the peaceful weekend ended in an instant.
Then, as I went to light the stove, that was when the fire just consumed me.
The alcohol stove had somehow misfired, exploding and instantly engulfing Catherine and the boat's galley in flames.
I heard my wife screaming, and that alerted me. I woke up and turned and saw her all aflame. Her hair, her face, her clothing, all on fire.
The first thing I screamed was, "Help, I'm burning. Cyril, wake up, you gotta help me, I'm gonna die."
She was engulfed in flame. And I just, I didn't think at all, I just reacted. I was just gonna put the flame out. And I did that as quickly as it could be done.
Cyril put out the fire with a blanket, burning his arms and legs from the intense heat in the process. Fortunately, within minutes of the explosion, a Coast Guard cutter arrived. Catherine and Cyril were airlifted to the nearest burn trauma center, Brigham and Women's Hospital in Boston, where Catherine had worked as a nurse many years earlier.
I could feel myself being moved through doors.
She had third-degree burns on her arms and second-degree burns on her face. third-degree burns completely destroy all the layers of skin and cause severe scarring. While second-degree burns also leave terrible scars, not all the skin cells are killed. Those few remaining cells were Catherine's only hope of getting back the face she had come to recognize as her own.
We photographed them immediately and then photographed their progress on it.
It would be up to Dr. Robert Demling, director of the Burn Trauma Center at Brigham and Women's Hospital, to save Catherine's face. He said he knew it would not be easy.
This is Catherine on admission, and this whitish area on the lower part of her face involving her nose and her neck is deep burn. And this char on her forehead is also deep burn. It's dead surface skin. She has a tube in her mouth because she's on a ventilator at this point in time.
The ventilator kept Catherine from suffocating. Her burns were so bad and her swelling so severe that her air passage was in danger of being squeezed shut from the swollen tissue. Dr. Demling quickly stabilized Catherine, and then he turned his attention to her scarring.
Don't worry about that. Once you get a burn, your face, particularly a facial burn, your features are really distorted, especially from the swelling and the color change. So it'd be hard to even recognize your own relatives.
Cyril's burns were fairly minor, but he was still in pain. He couldn't lose the image of seeing his wife's face on fire, and he couldn't help thinking about what she might look like once she recovered.
I thought she'd be a mess afterwards. And
Demling knew there was only one way to keep Catherine's face from being scarred beyond recognition, and it was the patch of skin being developed at a company called Advanced Tissue Sciences. Researchers at the company came up with a way to create human skin. But before skin can be grown, it has to have a seed, a tiny bit of natural material that starts the growth.
The product lifts off.
This modern skin comes from an ancient religious and medical practice. It comes from the discarded foreskins of circumcised baby boys.
So we take these surgical discards, enzymatically treat the tissue to remove the cells, and then start our cell banks. And it's really remarkable because these are young, healthy cells that have a tremendous potential to grow. And from one starting material, we can actually get 250,000 square feet of final product.
That's 5 football fields of new skin from one tiny foreskin. As thin as a paper towel, biodegradable skin is the first FDA-approved tissue-regenerated product. Dr. Demling thought it could save Catherine's face. Not only would the skin eliminate scarring, but it would eliminate something else that has always been part of burn treatment: pain.
Which is very different than traditional care, which requires painful wound care dressings 2-3 times a day for several days, so painful that morphine has to be given to the patients.
Until now, doctors have used skin from animals or cadavers to cover severe burns. But human immune systems react violently to both. However, the skin flap eliminates the possibility of rejection because of the way it is made.
The cells themselves are surrounded by a naturally secreted matrix. So they basically are coated by collagen and other naturally secreted proteins, which are not different from one person to another. So what the immune system sees are not the cells, but the matrix that surrounds them sees that as self and doesn't reject them.
And then on day 8, we can see most of
The laboratory-grown skin would remain on Catherine's face while her own skin grew underneath. In only a week, the patches peeled off, almost like a bandage. The results are immediately apparent.
You can see the smooth new skin on her face now.
The miracle skin did save Catherine's face, but her arms were too severely burned for the skin patch.
Tissue regeneration saved my psychic. I'm so scarred in my arms, but my face is fine. And that's because within a couple of hours I was in Brigham and Women's, and a few hours after that, I'd had the treatment.
Doctors say Catherine's face is remarkably radiant and completely unscarred. If they had treated her without the regenerating effects of the skin patch, that would not have been the case.
She really has no scar today, and I would have predicted more scar on her face if we would have used the standard treatment.
Thanks to modern medicine's ability to regenerate life, Catherine's radiant smile is shining again.
It's my whole world, my whole being, my whole ability to go out and face the world and go back to work as quickly as I did, be able to function as quickly as I did, to be able to sit here on the boat, was all because of the technology.
Early success with growing skin for burn patients has led scientists to explore the possibility of growing even thicker tissues in the laboratory. Tissues as thick as human cartilage. The pain from torn, damaged, and dead cartilage is a curse to a young athlete like Morgan Montgomery. He suffers from osteochondritis, a wearing down of his cartilage, and after 4 knee operations, only temporary relief was in sight. He began riding a bike to help his cartilage, and he liked it so much he began cycling competitively. But his knee was not getting better. He pedaled on despite the pain. Then he learned about a procedure that could replace his cartilage, get him back on track, and put him in the race for a spot on the U.S. Olympic team.
Oh, it feels great. Not one problem. Feels just like a normal knee.
Last year, Morgan underwent a Carticel cartilage replacement procedure. Healthy cartilage cells from his good knee were grown in a lab and then inserted into his damaged knee.
If I hadn't had this Carticel procedure, I wouldn't be doing this right now. No way.
Today, Morgan's racing in the EDS Cup, a national race in his home state of Minnesota. If he wins, he'll be another step closer to making the Olympic team.
120 laps, 30 kilometers. For category 1, 2, and 3 points race.
In North Carolina, Kevin Ketchie is about to have the same procedure. Ever since he tore the cartilage in his left knee playing basketball, he can only sit on the sidelines and watch. Sometimes, that hurts almost as much as his knee.
I can pretty much keep my mind off of it if I stay away from here and just don't think about it too much. But you could tell when I came in here, it's kind of hard for me to just kind of sit around. I still want to be out there. So.
But Kevin hasn't taken a shot since the night his knee gave out.
I finished playing that game, actually. Did pretty well. Went home the next morning. I knew that I had really done something to it at that point.
Kevin faces a daily reminder of that night every morning when he goes to work.
I have a hard time at work just because of the fact that working at an old courthouse doesn't have an elevator. And my office is on the 3rd floor, which I really don't have a hard time getting up the stairs to the office. Coming downstairs takes a little longer just because of the way the knee moves.
It could be a prediction of what lies ahead, a lifetime of pain. Doctors told Kevin that sooner or later he would have to have the knee replaced with a mechanical one. More than 200,000 knee replacements are performed every year. And they work great, but they don't last forever. Kevin's mother is a nurse, and she told him that if he got a new knee at 28, he would need another one at 43. That was enough for Kevin.
If my mother wasn't a nurse, I probably would have went ahead and had the procedure done here in town with the first physician I went to, but she thought it was better through all of her experience to go ahead and get a second opinion.
So Kevin headed from his home in Greensboro to Duke University in Durham, North Carolina. He sought the advice of orthopedic surgeon Larry Higgins. Instead of a knee replacement, Dr. Higgins told Kevin that he could have a Carticel cartilage implant.
This procedure isn't for everyone, it's clear. It has very limited indications. This is not a treatment for arthritis. This is a treatment for someone who has loss of cartilage in a focal area on the femur bone and has an intact, the remainder of the knee ligaments and the remainder of the cartilage ligaments are intact.
The first step was to remove healthy cartilage cells from Kevin's uninjured right knee. The cells were shipped to Genzyme Tissue Repair in Cambridge, Massachusetts, and injected with growth nutrients, a sort of fertilizer for human cells. To repair Kevin's knee, Dr. Higgins will need about 12 million healthy cartilage cells.
Along with the basketball.
Just a month later, the new cells for Kevin's knee are ready. So Kevin and his mom return to Duke, where Kevin sees his restored cartilage.
I think I saw him bring the cells in the box a while ago. So a guy coming in with a box had Carticel written on the side of it. And my mom, of course, was like, well, there's your stuff.
That sheet goes just up on top.
Once Kevin is in the operating room, Dr. Higgins wraps his leg in an elastic bandage.
Leg. And that will allow us to minimize our blood loss and allow us to implant the cartilage in a bloodless field, which is the ideal growing conditions for the new cartilage we're going to implant.
Dr. Higgins can now open up Kevin's knee and look directly at the joint. The cartilage cells taken from Kevin's right knee are ready to be injected into his left.
Pull this out. And these are Kevin's cells all suspended in the fluid there. And we're going to take these, and then I'm going to slowly inject Kevin's cells into the little patch that we made for him.
All Dr. Higgins has to do now is close up Kevin's knee.
And that's the end.
And because the local anesthetic was used, Kevin hears the results of his surgery while still on the operating table.
Kevin, everything went great. Implantation went perfect. So, good news.
That's good news for Kevin and for thousands of people who injure the cartilage in their knees every year. He'll be on crutches for about 10 weeks and then start rehab.
By a year, he'll be released to full activities, playing one-on-one basketball, being able to compete at whatever level he wants to compete at. And that's really the purpose for doing this procedure.
How's the knee feeling? Oh, it feels great.
If Kevin had any doubts about the operation, all he has to do is look at cyclist Morgan Montgomery. Thanks to modern medicine's ability to regrow cartilage, Morgan is not only back in the race, he's winning. Morgan Montgomery is once again pointing towards the Olympics, something that wouldn't have been possible without the Carticel implant. From growing healthy cartilage and restoring dreams, doctors moved to the greater challenge of growing body parts.
So I'm going to go in and...
Regrowing bones wasn't the first thing on Dr. Joseph Vacanti's mind when he decided to get into tissue engineering. The field didn't even exist. Eventually, he would grow bones and much more. But in his early years as a pediatric transplant surgeon at Children's Hospital in Boston, all he knew was that the children he saw were dying, and he wanted to do something about it.
When I completed my training and the problem of organ shortage became my problem, then I decided, well, maybe the thing to do is just make organs. And then we wouldn't have to rely on a supply that would never meet the need. And so with those beginnings in the 1980s, with my good friend Professor Langer, we began down a road to try to understand what the problems might be, and then go on to try to actually design and build a whole organ.
A pretty lofty goal. But his colleague Bob Langer, an MIT chemical engineer, was no ordinary professor. And his specialty was building dissolvable plastic structures that harmlessly disappear after giving new cells a place to grow, or as they are called in the world of tissue engineering, biodegradable polymer scaffolds. Soon Vacanti and Langer were onto something. But what actually propelled them forward didn't come from a lab. It came from the sea. It was 1986, and Dr. Vacanti was on vacation in Cape Cod. He had never stopped pondering how to build a liver. As he sat on the jetty watching his family play, he saw some seaweed. A major breakthrough in tissue engineering was about to happen.
I could see seaweed waving through the water. As the water moved, so did the seaweed. And so it struck me that the seaweed was answering that question.
Dr. Vacanti's question was how to keep alive and grow the cells of tissue like cartilage and eventually the liver. The answer was in the shape of the seaweed, which allowed nutrients to reach a large number of cells deep inside and on its surface. The branches of seaweed were the key.
The fundamental repeating unit in nature for all living systems that are multicellular are branching systems.
Like seaweed. Dr. Vacanti immediately phoned Professor Langer, and they began working on a dissolvable structure that would look and act like the seaweed, branching out and supporting life. Before long, they were seeding their dissolvable plastic scaffolds with living cells.
This did exactly as the seaweed in the water did, and it allowed the fluids to percolate through it and keep much larger, thicker masses of cells alive while they formed new tissue and laid down their own matrix.
When they put these cell factories into bodies, blood vessels would grow into them, providing even more support. And eventually, the plastic or polymer scaffolds would harmlessly go away. Soon, Dr. Vacanti's brother, Dr. Charles Vacanti, an anesthesiologist, joined the team. Earlier in his career, Charles had witnessed a patient die from a tumor on his windpipe.
If this patient walked into our institution today, we have the science and the ability to resect that particular segment of windpipe and that tumor, and then to replace it with a tissue-engineered windpipe.
By the early 1990s, the Vacantis and Professor Langer were growing cartilage. It was significant research, but at the time, few people were paying attention.
As we did in this
so they decided to grow something that needed cartilage that no one had been able to create, an ear.
So we decided we talked to a plastic surgeon, Dr. Upton at the Beth Israel Hospital, and he said that the gold standard for a plastic surgeon was to try to recreate the shape of a human ear. He said, with all the technology that plastic surgeons had access to today, they still were not able to do that. So we said, let's sit down and generate cartilage in the shape of a human ear.
They needed help creating the mold that would dissolve harmlessly.
Well, we'd have the peptide solution, we'd have water. Okay.
So they solicited the help of Dr. Linda Griffith, an MIT chemical engineer who had come to Boston to work in the lab with Joseph Vacanti and Robert Langer.
And Charles had done this really fabulous work showing that you could combine cartilage cells with a polymer and get a piece of tissue to form, but lacked the ability to make that polymer in a specific shape. So they called me because they knew that I knew something about polymer processing.
In 1992, they presented their ear to the world on the back of this mouse. They had taken another step toward realizing a dream of growing human parts and organs. But the Vacantis' work didn't stop there. In 1998, Charles helped Raul Murcia, a 36-year-old machinist, regrow a thumb.
He was a laborer and working with industrial machine had chopped off the end of his thumb.
Once again, the inspiration for an innovative solution would come from the sea.
We've actually used 3 materials from the sea. The first is the seaweed for structure. Second, coincidentally, the hydrogels that we use when we make a minimally invasive implant, such as an injectable tissue, is derived chemically from seaweed. And third, when we made the thumb implant for Mr. Murcia, the base of the scaffolding was coral, which was derived from the sea, and then injected with this seaweed derivative referred to as alginic acid, containing his own bone cells.
Specially treated coral was used because it's very porous and would provide a nice template for bone growth.
It's chemically very close to the matrix of bone. So the chemical content of coral is very similar to the chemical content of bone minus the living cells.
Over time, the coral would break down naturally. Full regeneration takes 16 weeks. Week 1 makes sure there is no rejection. At week 10, immature bone growth can already be seen. During weeks 12 to 14, there's new mature bone growth. And finally, by week 16, ligaments and tendons are reattached, giving Raul Murcia almost normal use of his thumb.
He can lift very heavy objects, and it's fixed in this position, but it works exceedingly well.
Fortunately, he can now give the procedure a thumbs up.
Bueno, pues yo me sentí contento, ¿no?
He was very happy.
Dr. Vacanti will tell you there are many approaches to regenerating organs. If you visit Dr. Lola Reed's laboratory at the University of North Carolina, Chapel Hill, you'll find the possibility of regenerating livers.
Would you please get out one of the bags of the human cells?
Dr. Reed hopes to eliminate the need for most liver transplants with non-invasive liver cell therapy.
No, keep it on, keep it on.
She believes she can do this by growing livers in her lab.
To realize that there are special cells within the organ called stem cells, which are cells that are like seeds that can in fact flourish and grow and form the whole organ.
Dr. Reed and her associates are the first to figure out how to purify these stem cells in bulk and use them to regenerate a liver. With help from researchers at Albert Einstein College of Medicine in New York, they have shown that the cells can form liver tissue. They would also be available whenever needed, something that could end up helping millions of people with liver damage, including Maria Luisa Lujan, who lives in New Mexico.
I want to meet in sync.
The only sign that Maria is sick is the slight yellowing of her eyes and face, reminders of her rare liver disease. Doctors first thought Maria had neonatal jaundice, which is common in newborns. It usually repairs itself after treatment under ultraviolet lights. But Maria never improved. She was later diagnosed with Crigler-Najjar syndrome. Her liver doesn't properly excrete wastes or bilirubin. Maria is one of only 150 people in the world with the life-threatening disease. Maria says she never worries about her illness, but the same cannot be said of her father.
Unless we find a cure, we don't know how long we'll have her. But there's a lot of other parents, at least 150 that I know of, that have children that do the same thing we do. Care for their children and take the best care of them they can.
That includes turning on Maria's UV lights every night, a much larger version of the Billy lights that doctors first put Maria under when she was a baby. The heat from the lamps helps her sweat out the bilirubin her liver cannot eliminate. Without the lights, Maria could develop brain damage. Maria has never known life any other way, but her father says having a daughter who has to spend every night under these giant lights can take its toll on the family.
It's a devastating disease in more ways than one. You know, it really takes its toll on the body and the person and the people and the family. Like I say, you just can't get up and go on a vacation, can't go camping for 2-3 days. We haven't had a vacation since she was 3 years old. We just can't do it.
Before Maria becomes an adult, she will need a liver transplant. That will make her one of nearly 14,000 Americans waiting for a new liver. But only 4,500 livers will be available. Odds that trouble Maria's father.
She's my little girl. She's the only one she's my only daughter. And I guess it's just the father instinct to want to do more for your daughter.
Lujan searched the internet and found Dr. Ira Fox at the University of Nebraska. Fox was one of a few surgeons using adult liver cells harvested from cadavers to regenerate the livers of patients like Maria. But the new method still had the same old problem: not enough livers and too many people who needed them. Finally, after 2 years of waiting, Maria's liver was infused with 7.5 billion healthy liver cells. The treatment worked, and Maria can now go as long as 2 nights in a row without sleeping under the lamps.
She had rosy cheeks, and she looked really, really good. Her level had been the lowest it had ever been in her life. So we knew that there was some hope and there was something working.
Maria had to wait 2 years for her cell infusion, but patients in the future may not have to. Dr. Reed hopes to overcome the liver shortage by taking the cells of one liver and giving them to hundreds of patients.
That means that we will be able to stockpile the cells in a tissue bank and be able to ship them at any time. So she wouldn't have to wait in the future if we make use of the progenitor cell therapy.
Only the progenitor cells, the young cells, can regenerate. So they hold the key to turning a damaged liver into a healthy one. Dr. Reed shoots the progenitor cells with a laser beam, which colors them and makes them easy to identify.
And then there's a computer associated with it that can be told to take all the cells of a given color, let's say a yellow and orange ones, and put them into a particular test tube. And they will vibrate the solution to go to that test tube.
Finally, the cells are frozen with liquid nitrogen.
Because we will be able to store these cells frozen, it means that we can have lots of different types of donors with marker studies done on them, and therefore we can match precisely a set of cells that would go into a given patient.
The regenerated liver would have another advantage over the current treatment. Despite her progress, Maria still needs to take immunosuppressant drugs 3 times a day. Dr. Reed says this too would be eliminated.
So that means that that patient will basically be able to have a normal liver, normal function within a couple of weeks.
Let me just cast it in and she can... Okay, well, let me...
The cell infusion has helped, but Maria has not been cured. The cells from the adult liver will not live forever, and no one knows exactly how long they will last. If Maria's health is threatened by rising bilirubin levels, Dr. Reed's cell therapy is her greatest hope. It means Maria wouldn't have to wait for a donor. She wouldn't need immunosuppressant drugs, and her family would never have to worry about her bilirubin levels again.
It's going to revolutionize all of transplant surgery. Indeed, what is going to happen is that many of the transplant surgery that's done today will not be done in the future. Do a lot of homework.
Dr. Reed predicts that the liver will be the first organ recreated in the lab to reach patients, but that will only be the beginning.
Into a separate tube. The liver is going to be the first organ that is going to be done for cell therapy, but we think that all of the organs are going to be treated in this way in the future.
More than 40,000 people a year suffer second-degree burns. But to see Catherine O'Leary's face, you'd never know she is one of them. Morgan Montgomery continues to train for the Olympics and rarely thinks about his knee or the lab-grown cartilage inside it. And Kevin Ketchy can look forward to an active life, free from pain. But even the modern procedures they underwent may soon be outdated. The Vacantes are working on projects that seem too spectacular to believe. Joseph Vacante hopes to grow an artery inside a human heart, and then an entire heart. Skin and bones, cartilage and organs, even hearts, brains, and spinal cords, all will be created in a laboratory. Testament to modern medicine's ability to regenerate life.